obviously I have no interest in the manufacture of genes. On our staff is Dr. Sundararajan, who's at Aravind, Madurai, and does a lot of work in the genetics of glaucoma. Then we have Dr. Sharma, who's also the chairperson for the secretary of all the meeting here, and she'll talk on corneal and refractive surgery. We have Dr. Tandon, who will talk on the cornea. She's in charge of the eye bank. And we also have Dr. Sundararajan, who's very interested and does a lot of work in the genetics of the retina. And as our special guest, we have Professor James Elder from Australia, who is interested in pediatric ophthalmology, especially the retina. So let's start. And genetics has always been of interest. The special journal we have was in the various eye journals, archives over the years. You can see the talk. And also it hit the general population journals like Newsweek, Time magazine, and you want to know my future? Right there, your genetics. Now, genetics in the eye has at least 90% of the genes in the human genome that's expressed in the eye. So therefore, when we're studying the genetics of the eye, we're really talking about the genetics of the body. Here's a list of the genes that affect the eye. It's amazing. You can see so many of the general syndromes that have an ophthalmology overlap, like Marfan's, Stargardt's disease, of course, for the retina. So this is our talk for me this morning. I'm going to review genetics. I'm going to talk about current gene therapy and proposed future gene therapy. On top of that, there is an elephant in the room: artificial intelligence. So here's our crash course, Genetics 101, just to refresh your memories. We can see that the gene is mostly introns, and it's the exons that we're more interested in. It's interesting that the difference from one human to another is just 2% base of 1%, tenth of a percent of the base pairs. But for a human to a chimpanzee, it's just 2%. We're very close to a chimpanzee. So here's the spread of the chromosomes. The largest ones start at number 1, and the smaller ones at the end, except for number 21 and 22. They switch over. And here's a review of the chromosome. In the center is the centromere, at the ends of the telomeres, and they're numbered as you go up for their locations. And we know about meiosis and the crossover and how this is passed on and broken down. Of course, if it occurs in the same body, it makes no difference in your proteins because all the genes and DNA are there. But if it's passed on to another person, then that change takes place, because here we have translocation of part of the chromosomes to a chromosome 4. In the same body, it makes no difference, but if it's passed on to your offspring in meiosis, it does. Let's talk about DNA, very interesting, and the RNA. The base is a 5-carbon sugar. It's a pentose. We have deoxyribose, where there's a hydrogen atom assigned to carbon 2, and if we have oxygen, then it's ribose. Attached to that is the base, and we'll talk about that to number 1, and the phosphates on number 5. So therefore, a nucleoside becomes a nucleotide when you have the phosphorus attached to it. So here, this is it. We have 2 purines, bases, adenine and guanine, and you see why they're that way, and 2 pyrimidines, thymine and cytosine. here we are. And they're color-coded because when we do chromatography, these are the colors that we use. Now take a look at here. We could see why you cannot have two purines or two pyrimides hook up with each other. You could see in the bases the side. It's very fat on the purines, so very fat on those bases, so they can't hook up. So therefore, a purine could hook up with a pyrimidine that's thinner, so there's more room. And of course, they are tied together with the H bonds with the nitrogen. And therefore, we then get our codons, et cetera, and our genes. When we talk about DNA and the hookup, this is what we have. It first came out in 1953. Watson and Crick formulated the structure, unbelievable, with diffraction, X-ray diffraction. And they wrote about it, and then they followed a couple of months later with more of a description. And this is what we have today from Watson and Crick. Here's their talk, that the hydrogen bonds are broken, they separate, and then you could get replication. All this they figured out. Of course, they got the Nobel Prize for it. The DNA is compressed around little histones and then shoved into a chromosome in various locations. The exons are what's important. The introns got rid of it. Transcription is the elimination of the introns, and then we get the messenger RNA. And that messenger RNA, through translation, becomes an amino acid and ultimately a protein. And that takes place outside of the nucleus in the cytoplasm. And we can see it's like little plugs plugged in there. First the amino acids come and then the protein. And here's the mitochondria from ancient bacteria. And the DNA is similar to that of bacteria. It also produces the RNA and the protein. And the energy is really, in the mitochondria, adenine triphosphate and the Krebs cycle. If from back to medical school, you remember that. So here we can see the transformation of that. In the mitochondria, we have 37 genes. That's it compared to the nucleus. And here's a picture of it with its surrounding and ribosomes and the breakdown of the membranes and the energy areas. So for the mitochondria DNA, I said there's 37, but for ophthalmology, just 3 of them, just 3. Leber's hereditary optic neuropathy, Kern-Sir, and for the nuclear DNA, about 98. And hereditary is from both parents. So here's to break down the DNA, the sense, and then the opposite, the codons, the antisense, and then we have the translation into amino acids, and finally, the protein. So let's talk about the open reading frame, because it's 3-letter codons based upon the DNA or the RNA. The first letter makes no sense, but then you start with the second one in threesome. So forget the F. The cow and cat ate cod, et cetera, and then you have the end. It makes no sense if you start on the first letter. And this is the result. The amino acids, various codines, various combinations can make up the same amino acid. And it starts with a start codon, that's the start codon, AUG, methionine, and it ends with 3 of the combinations, the stop codons, and with that, the process for protein stops. But we can see how mutations can take place. We have a frame shift in which an extra base is inserted or deleted, and that changes everything. And with that, you can get various changes in the protein and diseases like sickle cell and hemolytic anemia by putting in an extra or changing the codon. And finally, the stop codon. And any single base or deletion could result in a stop codon, and that's the end of that amino acid sequence, and then you get a non-functional protein. And here's some of the associate genetic diseases, including retinitis pigmentosa. So we get rid of the junk introns, and at the end we have the ENCODE that will continue on into the human genome and who we are. We have epigenetics. I want to talk about epi and over. This is over and above the regular genetics, and you'll see that in a minute. Darwin talked about the evolution and epigenetics, and Lamarck was based on that, because we can see that evolution is a result of some genetic changes in a population, and epigenetics is expression over and above that. It could be inherited, but it's not in the DNA, as I'll show you in a minute. We have various markers that are affected, the methyl group and the acetyl group. Therefore, we can see that parts of our body stay the same. Here we have it. Histones, our DNA is wrapped around it, and when the strand is wrapped around a histone, it cannot be touched. But epigenetic mechanisms, drugs, aging, diet, environmental, that can affect it, but it's where it's open. In the lower right, it says DNA accessible, gene is active, and that's where we have the methyl, and that is affected and can be carried out. But if it hits it on a histone, it's not altered. It's just when that tail, the DNA is accessible. It doesn't change the DNA, but it hooks onto it. So let's talk about what genetics are. It's the study of specific genes in the DNA and RNA and inheritance and mutations. But genomics is the study of all the body's genes, the whole genome sequencing, so we could have a better understanding of genetic diseases. We can see that a gene is attached in a certain area of the chromosome, as I showed you before. It's wrapped up, spiraled, and put in there. So in molecular therapeutics, we talk about genome sequencing. We want to talk about a mutant gene and DNA replacement, engineering, etc. And for the study of it, at first we were using the polymerase chain reaction. We extracted the tissue, put it in a heater to break it down, and amplified it with a cycler. And this is what I was first doing at Tony Aldavi's lab for cornea. And this is, we could see how it breaks down with the heating. It could replicate, and we could then read it according to the colors. But now it's more sophisticated. We have newer technologies, faster, higher volume, and this is at Cornell, where I'm from. And this is, at that time, a sequencer that they had. And I'm also from San Diego, and there we have Singular Genomics. And this is a brand new high-speed sequencing that leaves nothing out. Very fast analysis of the whole genome, where the DNA and the chromosomes are. It's fast readout with a small piece of tissue. And that factory, that plant is on the campus of University of California, San Diego. And here I am at the plant with the sequencer. Not a big machine, but very effective. There's home testing kits, you could read them. By the way, 23andMe is now bankrupt, out of business. Others have taken over. You can see Illumina and Singular at the bottom. Illumina is also on the campus, or near the campus of University of California, San Diego. So now we talk about the naming of genes. The Hugo gene was set up because so many researchers, when they have a gene, they put a name on it, and there was no consistency. So this organization was set up to name the genes as we knew them, 19,000 of them. Even so, they have to convince the researchers to continue the proper naming of the genes. Craig Venter, it says Boston, Massachusetts, but his lab is also adjacent to UCSD. And it's very interesting because with him, it's like a computer. You change the structure, you change the input, and you get a different output, just like with the computer. When you change the DNA software, you change the species. Just like with the computer, you change the ones and the zeros, and you get a different readout. So here we can see a computer binary code and the genetic code. You change it, you get something different. And this is the work that's being done in his lab. He has one Nobel Prize winner there, too. So let's talk about gene therapy. We have vectors, they're usually the viruses, and they're altered to carry the human DNA. And we have to manipulate the viral genome to remove disease-causing genes and insert a therapeutic one, or do something with it. So use of viruses, yeah, that's the most common. Gene delivery puts a strand of DNA in a virus because we know viruses could affect the body, and we just hope that it goes to the right place in that huge genome. Herpes simplex is actually used practically, you can see, for dystrophic epidermolysis bullosa, a cloudy cornea because of defective collagen. And this work was done at Bascom Palmer in Miami by Dr. Sabata. He removed the scarring and then put the drops of the herpes on top of it with replacement genes to replace the genetics of the collagen in the cornea, and he got clear corneas. Right there, of course, cornea is on the surface and easy to get to. I propose using the coronavirus. After all, that hits everything and it keeps changing. We can see the spikes there, and you change some of those spikes, put some proper genes in there for replacement on those spikes, that could be easily carried into the body. They go into your body anyway, so let's control it. So we can see the fusion machinery and how it penetrates and goes through the membranes and into the place that you want it. We do have to aim it at the proper place, but with the coronavirus, the whole body changes. You could change the genetics of the whole body to what you want. Here's some of the vectors. The most common is the adeno-associated virus, and number 2 is the favorite for carrying viruses or altered into the body. We can see how this is a gene delivery tool. Take out the offending part, replace it, and then with that AAV, it breaks through the membranes of the cell, travels through the cytoplasm, aiming at the nucleus, penetrates the nucleus membrane, and there you are, it's altered. A new protein. There's other methods, non-genetic. Electroporation, high-intensity electric fields, short pulses that could make the plasma membrane of various ocular tissues change. And iontophoresis, that's low voltage, continuous, and you could get the permeability of the small nuclei. It could affect us in what we do. Now, if we're going into space, like the couple did for supposed to be 1 week, it was 9 months. They're exposed to galactic cosmic radiation and solar particle events. Their DNA, the strands break, the cell cycles arrest, and the cornea, the stromal edema and epithelial thickening. This was done in mice, and it could be done in them too. And they're being examined for that upon their return, finally, after 9 months. We're talking about CRISPR. Now, Jennifer Duda was instrumental in the consideration of CRISPR. She got the Nobel Prize for it. So it's clustered regularly, interspersed, short palindromic repeats. I'll ask all of you at the end if you could repeat that. And it works with the Cas9, which is the enzyme that will break the tissue, the cutting edge enzyme for the DNA, and therefore we could replace certain sections. Again, it's the clustered regularly interspaced short palindromic repeats. And remember that a palindromic repeat is a double-stranded sequence of DNA. The order of the nucleotides are the same on each strand, but in opposite directions. So in effect, it's an antisense. And the CRISPR will break it into pieces, plasmids or viruses, and it's molecular scissors to cut it. It cuts a genome at a specific place, like this, and can replace it. That's CRISPR. Now, of course, it's controversial because if it's done with embryos, you could probably not only cure potential genomic anomalies, but also you could have social Darwinism by gene modification. You could plan for a super athlete or a brilliant genius. Well, that's what it is. Finally, I want to talk about AI. At Cornell, I was with a plant geneticist in a fellowship, and we talked about AI because they're programs, models that could really revolutionize how we think and see biology and how we could predict the genomics of plants, in her case, and humans in us. What's cool about this is all they need is the input of the DNA sequence, and they could take over after that. However, Eye magazine from the Royal College of Ophthalmology a few months ago talked about threats in academia, evaluating plagiarism and artificial intelligence with ChatGPT, and this is from an ophthalmology journal. And I had this situation at Cornell with the professors who told me that they get beautiful readouts, papers from their fellows who speak poor English, but the paper is perfect. Artificial intelligence. So what he does is sits down with that fellow and asks them questions about the paper that they wrote. And I spoke to another professor who had the same problem. I told him what the first professor does, sits down and questions the student about what was written. Here are my references, and we're really off to the future. Thank you. Okay, I'd like to introduce Dr. Sundarasan, who will now speak on the genetics of glaucoma. He's a researcher. He's in Madurai at Aravind and has done spectacular work. And his daughter is now doing it too. Yes, sir.
Hello, can you display this? Okay, so very good morning. I think I'm not going to say the attendance is poor, but whoever is here, they are really interested in science, especially in the field of ophthalmology. I can take it that positive attitude, and I'm delivering the talk. Good morning. I'm Dr. Sundarasan. I'm heading the Department of Genetics. For the past 25 years, I'm working in the field of ophthalmic genetics. So you name any kind of disease from cornea to the optic nerve, we extensively studied, and then we published more than 160 papers, and I produced more than 25 PhDs, and really you are interested, please go through our publications. So lack of time, what I'm going to do what I did for the past 25 years in the field of glaucoma. So that's what I put the title, A Journey in Glaucoma Genetic Research. I think concerned with the glaucoma, the ciliary body secreting the aqueous humor, the aqueous humor, and then from the posterior chamber to anterior chamber, and then it is draining out to the abscissal vein through the TM and SEMS canal. And then there are so many other, the glaucoma is a irreversible blindness due to the dysfunction and the damage of the retinal ganglion cells. So once the extracellular matrix blocking the meshwork, it increases the intraocular pressure, and then the patients lose the peripheral vision. In addition, the restriction fluid movement in POAG or in the steroid-induced glaucoma, it is the reason because formation of the cross-linked actin network, abnormal development of extracellular matrix, and then decrease in phagocytosis. In addition, central corneal thickness, there are so many other reasons for formation of the glaucoma. So we extensively studied primary open angle glaucoma, primary angle closure glaucoma, pseudo-exfoliation, and primary open angle glaucoma. So concern with the myocilin gene, that is the first gene, is the candidate gene for primary open angle glaucoma, that is identified by the Dr. Stone group from University of Iowa in 1998. That time, the entire world thought about is the only one gene. Now the scenario is different. It seems to be is a very, very complex disease. Glaucoma is a very, very complex disease. Now they are telling instead of complex, it is an incredibly complex disease. That's what I am putting a pathway here. There are so many pathways are involved, and then it is very difficult to understand what exactly the pathogenesis. So you can see the yellow dots here, highlighted here. There are so many important factors associated with the primary open angle glaucoma. We are working on one particular dot to understand the pathogenesis of primary open angle glaucoma. So what we did for the past 25 years, we recruited huge number of primary open angle glaucoma patients, and then more than 500, and then we screened a specific gene that is called myocilin. You can see the exon 1, 2, 3. The exon 3 is the hot spot. There are more number of pathogenic variation we identified in the exon 3. And then what we did, when you observe a particular family, this is an autosomal dominant disease. Based on the volume of the file of the patients, we can able to say the severity of the disease. So one particular, lack of time, I'm going to focus on only one particular mutation in the myocilin, that is nothing but the glycine 367 arginine, meaning you and me, if the intraocular pressure is normal, that is we have glycine in the position of 367. So because if there is any mutation, it is instead of glycine, it will convert to arginine. So based on the change of the protein, what it will do, it will accumulate or it will interact with other normal and abnormal protein. It will form the extracellular matrix. That matrix is blocking the semscanol or the TM. So that's what we are getting the increase the intraocular pressure. So what we did, we have taken a vector that is called pcDNA3, that is a kind of vector, and then we cloned the entire myocilin gene, and then using the side-directed mutagenesis, we changed the glycine 367 to arginine. And then you can see the panel 1, 2, 3, untransfected, and then wild type, and then glycine 3, the mutant. Meaning, so the untransfected in the sense, we didn't put any DNA, but in the wild type in the sense, the wild type myocilin is transfected in the TM cell. This is the TM cell. What in our lab, we have International Eye Bank. So we'll take the TM and then we'll put it into the culture. We can make a primary culture derived from the TM. You take the TM and then you can do the experiment. That's what we are doing. So the wild type, the myocilin, and then you have to understand that Gerald talked about the gene and gene protein. So the expression of the protein is very, very important. Some of the protein, it will come out of the cell. Some protein will sit inside the cell, but the concern with the myocilin, it is a very leaky protein. Some proteins sit inside the cell, some proteins come out of the cell. If you measure the TM, then you can isolate you can detect the protein outside the cell or inside the cell. So you can see here the comparison between the wild type and then the mutant. You can see the green signal or exclusively the myocilin. The signal is more in the mutant than the wild type. Meaning we proved using the Western blot with a specific antibody. So you can see in the wild type, the intensity of the band is thick, both in the intracellular and extracellular, as I told you before. But in the mutant, you can see in the intracellular, the protein, the intensity is very high than the extracellular. Meaning all the protein is, once it is mutated, It is sitting inside the cell, it is not coming out of the cell. That's what you can see the intensity of the green signal in the wild type is very, very high. So due to this, the extracellular matrix, is blocking the mesh work, increase in intraocular pressure, peripheral vision loss. Okay, so now, I think you know about the liquid biopsy. The same way we have proved the control and cataract. During the surgery, just take the aqueous humor and then measure the protein. For your kind information, when you measure the aqueous humor, especially in the primary open glaucoma, the TGA beta 2 level is very high. But in addition to that, here the primary open glaucoma, see the intensity between the cataract. The cataract and POEG, the intensity is very high, meaning the amount of myocilin is very high in the primary open angle glaucoma patients. And then this is the long story. Once the gene is identified, the entire world is, very happy. It's only one gene. That time, we made a model. Nobody made the model for a computer model. So what we did with the collaboration with the Madurai Kamaraj University, Dr. Krishnaswamy, we made a face building model for the entire myocilin. That's what we did it. And then I told you before the TJ beta. So we are collaborating with all over the world, all the glaucoma groups, and we did the genome association studies or the target sequencing and everything. So here you can see I told you the content of the TJ beta is very high in the aqueous humor. Here genetically we proved that TJ beta receptor 3 is very significantly associated with the primary open glaucoma. I can give you another information about TJ beta. You can see, if you see the TJ beta 1, 2, 3, and then TJ beta receptor 1, 2, 3. Here, according to the genetic screening, we got more association in the TJ beta receptor, and then we published this paper in 2015 in Human Molecular Genetics. All the authors, I think they are the leading glaucoma groups all over the world. And then, interestingly, there is another gene that is called SIX6. This is another candidate gene. Now, for your kind information, myocilin is not only the one candidate gene. There are 4 candidate genes already identified. In addition to the candidate genes, more than 540 associated genes are there. It means that's what we are telling, it is incredibly complex. So this particular gene, we did the genome association studies. You can see the round. We got the hit in the SIX6 is one particular gene, is closely associated with the primary open glaucoma. Not only primary open glaucoma, it is also associated with the normal tension glaucoma. Then when we screened our cohort, when we screened our cohort, we identified lot of variations. Interestingly, in the promoter region, there are 4 base pair deletion is there. So that is a very, very interesting story. You can see this is the promoter, the enhancer sequence. I can put it here. You can see the ATCT. You can see it in the center. So that is nothing but the 4 base pair deletion is deleted in the particular patient. Now, if you look carefully in that particular region, there are more than 4 transcription binding domain is there. It means once the particular 4 base pair is deleted, lot of, it won't synthesize the right protein. So that's what, the severity of the disease is very high to this particular patient. So then what we did, again, we cloned the gene, we deleted the 4 base pair, and then we had to prove it in the GFRA-fish model. That is the biological relevance. What exactly you are doing in the bench, you have to prove it in the animal model. So here we have taken the GFRA-fish model. So this is the one. One is the wild type and then the mutant. We created 2. You can see, especially in the GFRA-fish, please observe the globe. The globe size is normal in the control, but in the mutant, the globe size is reduced. You can see in the panel F1, especially E and G. So the globe size is big in the normal, and then in the mutant the globe size is reduced. You can see the retina. So especially the ganglion cell layer, amacrine cell layer is very normal in the control, but here in the deletion one, it is completely, shrinked, or it is reduced the retinal thickness. So through that, we proved both genetically and then with the biological relevance with the zebrafish model. We published this paper in Molecular Genetics and Genomic Medicine. These are our collaborators. And then, interestingly, we supposed to make a DNA chip for diagnosis of glaucoma in India. So that's what, we made a collaboration all over India, the leading institution, and more than, we collected not only in our place, we collected north, south, east, we collaborated with all the leading institution, and then we collected more than 1,000 sample, and then we screened what is the exact the variation within the population. But because of the heterogeneity, the problem is the heterogeneity. Because of the heterogeneity, we can't be able to find out the common marker all over India. So now we are adopting another strategy, like you can recruit the patient, and then based on the intraocular pressure, meaning when the age is less than 35, if it is a juvenile open angle glaucoma, intraocular pressure is very high, definitely they are positive for myocillin. So like that, you can able to screen, and then we can go for counseling. That is the story. Gerald, I will take another 2 or 3 minutes, please. This is another family. It's a very, very interesting family. More than 5 generations, and then more than 400 study subjects. Out of the 450, 20 of them are primary open angle glaucoma, 20 of them are suspect. So we did the entire genome-wide association studies, or we screened the whole exome sequencing, and then we can't be able to identify any mutation in the known genes or the associated genes. That's what I told you before. 4 candidate genes for glaucoma, 450 genes are associated genes. We can't be able to identify any mutation. It means there may be an additional candidate gene for this particular family. Then again, we screened the through whole exome sequencing, both intronic and exonic region, we entirely screened. Now, the what we identified, that is called rare variants. The rare variant is associated with this particular family. That is the low frequency of the pathogenic variants in multiple genes is playing a role for this particular family. That is the ultimate outcome of this particular study. Now, this is, as I told you, this is a JOAG family. You can see is autosomal dominant disease. Is a vertical transmission, or any one member is affected in this particular generation. Obviously, this is the myocillin mutation because the intraocular pressure is very high, the onset is less than 35. Now, this is the current recommendation for JOAG and POAG. If it is a JOAG, onset before age 35 with the family history, with and without family history, we can go for myocillin. Or it is a primary open angle glaucoma, onset before 50 years, with the family history, we can go for myocillin. This is the recommendation worldwide to screen the myocillin. I think in addition to that, we extensively worked on angle closure glaucoma. We identified the candidate genes because angle closure glaucoma, there are 2 schools of thoughts. One is like anatomical problem. It is not a genetic problem, but the scenario is changing now. 60% of them is anatomical and 40% of them is genetics. So we recruited all the cases, and then again, it's a collaborative project. We collected 10,503 cases and then 29,567 controls all over the world in 20 countries, and then we identified the candidate gene for angle closure glaucoma. Not only candidate gene, the loci identified for angle closure glaucoma. Okay, this is the one. In addition, as I told you, if you are a clinician, please, whatever either the aqueous humor or vitreous humor tear is very, very important. All the biological fluid. Through that, you can find out. So what cytokine is correlated with the pathogenesis. That's what we did here, and then all the informations are here. Due to lack of time, you can read our publication in the current eye research. So I think very recently, I think we got a major grant from Indian Council of Medical Research. So my collaborator is Aparna Rao. So here we are making a kind of different strategy in the sense, so you have to take exactly where the pathogenesis is going on. You have to deal with that particular disease, tissue, then only you can able to understand the pathogenesis of any kind of disease. So here, our proposal is, what we proposed, suppose when you do the tuberculotomy, so I think is a kind of invasive procedure. You are not taking only the TM, you are taking along with the sclera. So when you grow it in the lab, the sclera cells is growing faster than the TM. So you can't be able to deal with, because our aim is to make a single cell RNA sequencing to find out what exactly the pathogenesis. So here, Dr. Aparna is very good in this particular surgery. She made a review article. And then she published that one. Anybody is interested, go through that. So ultimately, maybe I can present this particular data in the next meeting. So I think I have, collaboration concern with the glaucoma all over the world, and including in our hospital. I'm collaborating all the our glaucoma specialists with my students and I think. With this, I will stop here. And finally, this is my slide. Even I put a slide, again, I'm correlating with the science. You can see in the center, you can see the light, and then the periphery is, not that much. It means the glaucoma patients, they are losing the peripheral vision, they are getting only the central vision. So our aim is to, at least, why not you bring back the vision to that kind of patients through our research. Thank you very much for your attention. Thank you.
Thank you, Dr. Santarasan. While he's here, do any of you have any specific questions you'd like to ask in front of the group? And then afterwards, you could ask him questions privately. Okay, we're going to get on to our next speaker, who we're thrilled to have, and that's Dr. Adhika Tandon, who's going to be talking on the genetics of corneal dystrophies. This is a very important talk, so I'm glad that you're here.
Thank you so much, Jerry, for inviting me to participate in this instruction course. And genetics of corneal dystrophies, keeping in mind the theme about the current methods of genetic analysis and potential genetic treatments, I've tailored the talk according to that. And basically, the advances in ocular research is another frontier that has opened up: molecular genetics, epidemiological studies. And then when you go for interventional studies, you can have a holistic approach, a multidisciplinary approach, and also personalized medicine. And as you've seen before, the molecular genetics is giving you that platform that you can aspire to in future to give precision and personalized medicine. However, this is not very easy to do. There are a lot of funding initiatives that one has to generate. There are several platforms. In India, we have the DPT, DST, ICMR, CSIR, to name a few. And generally, with all research proposals, for some of the new people here, of course, you're already tuned to all this, that you have ethics, one can consider conflict of interest, finances. All of this also has to be taken care of by a PI. And people who are going to progress to be their PIs, themselves independently run a research project. It's important not only to do your project work, but also see how your superior, your head, your PI is managing things, and try to help in those protocols, because you are the future. By the time all this is going to translate into an actual event for a patient, it's going to be several decades for most diseases. So therefore, we're looking forward to the young ophthalmologists today to be interested in research and to take this forward. Now, what about cornea services and how are we placed in India? Now, we do have our unique populations, and a little perspective about how there's been a sea change and how one looks at corneal dystrophies. Originally, it was a very anatomically oriented classification system, but this has now been updated. It was initially proposed in 2008, then updated in 2014 and 15, and again further now it has been defined in 2024. So updated classification, what drove this change? The key change was genetics. It was only when the genetic information came through, it was clear that you could have the same gene and have different manifestations. Different diseases of the cornea could be affected by the same gene. And similarly, the people with the same gene would even have varied manifestations. So that was very confusing, and ultimately it is the genetic makeup which actually classifies the dystrophy and gives it its place. So there are various categories. As you can see, that there are some where one has not reached that level of having a very clear-cut identified locus and gene, and the ones which are doubtful have now been removed from the list. So I'd encourage you to please look up IC3DC. It's available open access through the Cornea Journal. And now, what implication does it have for India and Southeast Asia? So we have obviously our own problems, our own niche areas where we need to focus our energy. And we held this kind of a think tank workshop several years ago to try to identify that if we were to apply for funding together, what diseases should we work on? And that time we collectively decided that it would be retinitis pigmentosa and CHED. CHED is congenital hereditary endothelial dystrophy. So there was a lot of discussion, a lot of back and forth, and there was not only a question of what is important, but also what is doable, feasible, and what would make a sea change. And the idea is that one should have a consortium of organizations which are working for the study of the genetics of ophthalmic disorders in India. And then, of course, there was the Asian Eye Genetics Consortium, which was created. So a little perspective about now, a lot of work has been done on corneal dystrophies. I mean, a lot of publications from South India, but there was less information from the north of India. And we know that the corneal dystrophies have varying features. So we had wanted to screen our patients to see if it is mirroring the reported information from other parts, or is it unique? And there were certain unique features that came across. Basically, the distribution was a little bit different. The autosomal dominant, there were certain new mutations which were identified. All this information is published. So if you look up Paliwal, R. Tandon, and corneal dystrophies, you will get the publications to go through. And then so I'm just trying to show you in a snapshot that how the work was done and how one found novel changes. One found that there could be variations in clinical presentations. This was a novel mutation seen in granular corneal dystrophy, for example. Another novel mutation seen with Reese-Buckler corneal dystrophy and Avellino corneal dystrophy. If we hadn't started this journey, this would have been left and would never have come to the notice of the scientific community. So therefore, there is an encouragement that one should continue doing research and find the ways to get through the hurdles. Here again, there was another variation with lattice corneal dystrophy, a new mutation, another one in MAP-dot fingerprint dystrophy. And now the question is that does it help you in your clinical care? It definitely does, because now that we know what are the patterns, we are trying to get the routine screening for all the patients, plus we are able to have a good database and a good atlas of what the clinical pattern looks like. And of course, the holy grail is to have a very good correlation between the clinical picture and what is the likely gene. However, that is a little complicated because often you have patients in different stages of the disease who may come, and also their disease pattern may be complicated by different things like environmental factors and other things such as UV exposure. They may be associated pterygium, or they may be associated spheroidal degeneration of the cornea, and so on. So similarly for endothelial dystrophies, again, we have looked at our CHED cases. We had more males than females. 39% only were familial, and we had also looked at Fuchs and posterior polymorphous corneal dystrophy. And a certain new features we found because we've actually had patients of CHED who even 30 or 35 years old. That is simply because of the lifestyle of the patients. They were able to manage with that poor vision for so many years, which is unheard of normally. Like normally would expect the patient to present within 1 year of birth, and in fact at birth. So the way our country has progressed over years, of course now it's rarer and rarer to get patients presenting so late, but still it gave you an opportunity to see what is the natural cause of the disease. So you can see there was a Hudson-Starley line, there was spheroidal degeneration, and so on. In Fuchs endothelial dystrophy, again, there's scarring, Bully, and Desmond's folds. Most of these are very common changes which are seen internationally and elsewhere as well. And then we looked at, in CHED, the SLC4A11 gene, and out of 35 patients, what is intriguing to me is that though the patients were very characteristic, and there was no doubt that it was CHED, but we were able to find the mutation only about 39% of cases. So this is again work in progress as to what could be the reason for that. And it has a relatively lower occurrence in North India. The pattern of consanguinity seen in North is among cousins rather than between the uncle and the niece. Maybe, I'm not sure whether that makes a difference. And a high degree of consanguinity was seen among the patients of CHED. So no mutation was identified in 21 patients. They were screened. So what did we do with them? We screened for putative promoter. Again, no mutation. Screened for COL882 gene, whether it was there was a theory sometime earlier that maybe patients with Fuchs, some of their other relatives may have CHED, maybe it's a early onset, but that was not the case at all. There were very, very clear-cut clinical cases of CHED, and there could be existence of genetic heterogeneity, overlapping cases, and definitely lots more work to be done. A quick word about the molecular analysis for Fuchs dystrophy, COL882 gene. That is also something that we have worked on, and I'd like to acknowledge the Department of Biotechnology, Department of Molecular Genetics, led by Arundhati Sharma, the cornea services, all the clinical faculty, the ocular pathology, and the Department of Anatomy. So as I said, you look up Paliwal, Arundhati Sharma, Radhika Tandon et al., and you will get most of the publications if you're interested. Thank you so much.
Well, thank you. Thank you so much, Dr. Tanner. That was a very good summary of the corneal dystrophies. Now, we still have a few minutes. Does anyone have any questions for Dr. Tandem? Because Dr. Tandem has to go to another meeting. This is the time to ask questions. Well, we all know everything now. Very clear. I appreciate it so much. Thank you. Okay, I now have the honor of introducing our guest speaker from Australia, Dr. James Elder, who will talk to us on the genetics of familial exudative vitreoretinopathy and Norrie's disease. He's done a lot of work in pediatric ophthalmology and its genetics. So let me have the pleasure again of introducing to you Dr. James Elder.
Thank you. I'm very honoured to be here and somewhat humbled, actually, because I'm not really a geneticist, as most of my panels are. I'm a clinician, and this is a clinician's input on these groups of disorders. So first, a little bit of history. Norrie's disease, in retrospect, was first described by Gordon Norrie, who was a Danish ophthalmologist of Scottish descent, which explains why he's got a very Anglo-Saxon sounding name, but worked in Denmark. And he described a group of boys with congenital visual loss who, by the time he saw them, really had thusical eyes. It was Mette Warburg in the 60s who defined this condition more accurately and gave it the name Norrie disease in honour of Gordon Norrie. And in the mid-60s, she recognised that there was gross abnormalities with the development of the retina and retinal vessels, with proliferation of the retinal vessels. Creswick and Scapins in the late 60s described what we now all recognise as familial exudative vitreoretinopathy with exudative and tractional changes in the retina, and gave the name to this condition that we now recognise. And this condition, to clinicians here, will be well recognisable. In more severe cases, you can have total retinal detachments or retinal folds. In less severe cases, you have incomplete development of the retinal vascularization, and what, if you were looking at a premature baby, you would say is peripheral avascular retina. And these less severe abnormalities, if they're just observed, can sometimes progress, and so you can go from having an avascularized retina to a retina with tractional changes over time if you don't intervene. Prendergast and Tracy classified the various degrees and severities of the retinal changes in this condition. So, broadly, we recognise that there are phenotypes that are purely ocular, and the mutation in the Frizzle4 gene is an example of that. It's autosomal dominant, and it only has ocular features. And the archetype, or the typical prototype familial exudative vitreoretinopathy, which has systemic disorder, is Norrie's disease. And so some people with the Norrie's mutation will only have fever, others have systemic abnormalities, including hearing loss, intellectual disability, dementia, and behavioural problems, to name a few. The Canadian Fever Consortium published a paper recently, earlier this year, in which they looked at the genes that are associated with fever. The ones marked with a red star are part of the Norrin-Wnt signaling system, so that's Norrie's disease, and some of the fever-associated phenotypes with the other different genes. And then there's a whole host of other genes, and this is in fact even an incomplete list. I can think of a couple of conditions that are not on this list. Revesz syndrome is an example. And what this group suggested is that when you're doing sequencing, you should do a sort of a targeted approach. Start with mutation analysis, looking at things that are in the Norrin pathway, and then go a bit wider. And some of these are also recognised on the basis of the syndromes. Before going on to consider a little bit, need to consider some of the development of the inner retinal vascular system. Basically, this is a neuronal advancement from the optic nerve that drags the blood vessels behind it. And so a lot of the factors that are important for the subsequent development and angiogenesis are actually derived from neurons, the platelet-derived growth factors, VEGF, and importantly Norrin, which comes from Muller cells. This is a fairly old schema of the bits and pieces that are involved in driving vascular in retina, and the Norrie's pathway is on your extreme right, and it's interesting that exactly how it interfaces with this is somewhat uncertain, and there are a whole lot of other presumably undiscovered genes here, and I'll talk about that a little bit more. So the Norrin-Wnt signaling pathway is a very important pathway in embryogenesis, and Norrin is a protein in the retina that's secreted by the Muller cells. It interacts with a number of receptors on the cell surface, and they stabilise a intracellular protein B-catenin, which then passes into the nucleus and causes gene transcription. And I'll talk about the association of the severity of phenotype with different variations in this. Just to digress for a little bit to talk about Wnt signaling, this was first described in fruit flies, so these are very important secreted growth factors that are involved in embryonic development in all animal species and in most structures. The interesting thing here I discovered is that the original description of the wingless gene was here in New Delhi in the Institute of Agricultural Research a little over 50 years ago. R.P. Sharma was the scientist that described this, and this was the beginning of our understanding of this very important embryological pathway. So, in terms of genotype-phenotype correlation, in Norrie's disease, with the abnormalities in the Norrie disease protein, this is getting better understood. There was quite a nice meta-analysis published a couple of years ago now that looked at the published literature and in terms of the ocular phenotype divided it into mild, which would be peripheral avascular retina, moderate, in which there are significant exudative changes and the beginning of some tractional changes, and severe, in which there was total retinal detachment. Here's some examples of moderate and severe abnormalities. And what they showed is that there is a correlation between the severity of the retinal phenotype and indeed some of the systemic features, particularly hearing loss and intellectual disability, depending on what part of the Norrin protein is affected. And look, it's quite a detailed paper, but there are a couple of things. So if the effect is in the more peripheral binding site with the LRP5 receptor, then you don't get severe retinal phenotype. Whereas if it's in the cysteine, which is very important for the formation of this cysteine mutation, very important for the formation of the Norrin dimer, then you fairly universally get a severe retinal phenotype. So there is some emerging understanding of that. And the same thing applies, and this is just briefly, again in the phenotype with the associated features. Again, the peripheral part of it, of the binding, if it's with the LRP5, there are no systemic features, and so you tend to get, that's when you get the fever isolated pattern without systemic features. And it's a little bit variable with the others. You can see with the cysteine, rather than them all having significant systemic features, a significant number do. And so there are these emerging understandings of that. Now, where are all these missing genes? They may well be in the other parts of what I'll call the angiogenesis cascade or control systems, but Professor Sundran presented a paper in his presentation on enhancers and regulatory elements, and I think in fact that's probably where a lot of the remaining genetic discoveries with this condition needs to be. And one potential example is if you look in OMIM, exudative vitreoretinopathy type 3 doesn't have a gene associated with it, but its locus is very close to exudative vitreoretinopathy 1, which is the frizzled gene thing. So I do wonder as a clinician whether someone's going to identify a regulatory element there that controls the frizzled gene. And this is important. And the other example is nothing to do with fever, but I was recently involved in a paper with some colleagues from the US in looking at the cause of hereditary congenital facial paresis. And this is actually caused by a regulatory element that silences the GATA2 gene. And so, we are going to see more and more of this over time. And these regulatory elements are in the non-coding area. And so the junk DNA is perhaps not quite as much junk as we thought. I wanted to talk very briefly about treatments for these conditions. So in some respects, they're a little bit like severe retinopathy of prematurity. This is a child I saw with Norrie's disease who was born at 34 weeks, not because we knew this pregnancy was affected. The mother didn't seek antenatal advice. So am I allowed to go for a little bit longer? Yes?
He's asking for some.
Yeah, it's got a time up.
Take time, please, take your time.
Yeah,
Very
right.
important.
Yeah. So in this condition, this child was born at 34 weeks, and you really couldn't see much of the posterior pole because of extensive hemorrhage, but the peripheral retina did not appear to be vascularized, and it was a very low quality fluorescein angiogram we got because of the hemorrhage, but there were no retinal vessels beyond where the hemorrhage was. So this child was treated with bevacizumab, a VEGF inhibitor, and 4 weeks later, this is what we saw. The retinal hemorrhage had largely cleared. You could see that it was a very immature retinal vasculature with retinal vessels not really even getting to the fovea. And this is an OCT done at the stage that showed that there's gross perturbation of retinal development. You can see inner retinal layers, but they cease at the fovea, and it's just sort of unrecognisable as a normally developed retina, and this is at 38 weeks of gestational age. So the child then had extensive peripheral laser. This was just taken immediately after the laser, so you can see edematous retina. But all of this was to no avail, because this child had an examination under anaesthetic a couple of weeks ago, 8 years on, and this eye is NPL, the other eye is thicicle, so we were unable to arrest the process with this thing, and that's because there's so much disturbance of the angiogenesis pathways, and so even though the child had appropriate treatment, there are a few descriptions of this treatment being successful, but were probably with less severe mutations than this child had. So where are the other therapies going? From mouse studies, we know that if you have an ectopic source of norrin, so if the Norrie's protein, so if you have a knockout model in which norrin is not produced in the retina, but you engineer the mouse to have an ectopic source of norrin in the lens, the retina develops normally. So if we had some way of giving the child a supply of norrin, but it's not a once-off thing,